Have you ever wondered how scientists can find a single tiny virus in your body or solve a crime with just a speck of evidence? The secret is a revolutionary technique called polymerase chain reaction, or PCR. Let's break down what that means. Think of all the DNA in a living thing as a massive instruction manual with billions and billions of letters. Now, imagine you only need to study one specific paragraph from that entire book. How would you do it? That's where PCR comes in. It's like a magical, super fast photocopier that can find that one paragraph and make millions, or even billions, of copies in just a few hours. Let's use a simpler analogy. Say you have a favorite recipe, but it's hidden inside a giant cookbook with thousands of pages. PCR is like a special copy machine that knows exactly which recipe you want. It finds it, pulls it out, and starts duplicating it over and over again until you have enough copies to share with an entire stadium. So, how does this molecular photocopier work? It's a cycle that repeats itself. First, we heat the DNA. This causes the two strands of the DNA double helix to separate, kind of like opening the book so you can read both pages. Next, we add small pieces of DNA called primers. These are like sticky notes that mark the exact start and end of the recipe or gene that we want to copy. They tell the machine where to begin and where to stop. Then, a special enzyme, which you can think of as a little molecular librarian, hits to work. It reads the original strand and starts adding the correct letters to build a brand new, identical copy. Finally, we just repeat this whole process: heat, attach primers, copy. After about 30 cycles, what started as one single piece of DNA has been amplified into billions of copies. This might sound like a neat lab trick, but PCR has changed our world in countless ways. In medicine, it's the gold standard for detecting infections like COVID-19. It can take a tiny, almost undetectable trace of viral genetic material and copy it until there's enough to be easily identified. In forensics, a few skin cells or a single hair left at a crime scene can provide enough DNA to create a profile and identify a suspect. And in research, it allows scientists to isolate and study specific genes linked to diseases, helping us understand and fight them better. But it gets even more amazing. In today's world of precision medicine, PCR is helping us create treatments tailored to each individual. For example, in cancer therapy, doctors can use PCR to detect specific mutations in a tumor. This information allows them to choose a drug that is specifically designed to target that exact mutation, making the treatment far more effective. This is called companion diagnostics, where the PCR test acts as a partner to the therapy. It helps doctors decide if a patient will even respond to a certain drug before they start treatment. It's a huge leap from the old one-size-fits-all approach to a future where medicine is personalized to your unique genetic makeup. So, from diagnosing diseases to solving crimes and personalizing medicine, PCR is the powerful molecular photocopier that underpins so much of modern science. It's a simple concept with a world-changing impact. Thanks so much for watching. If you found this interesting, please give this video a thumbs up and subscribe to the channel for more science explained simply. See you in the next one.